we are going to start the new topic, which is in continuation to my earlier topic as a fresh lecture, is the extremophiles. Okay. What is extremophiles? As a word itself indicating, extremophiles means the bacteria which live in the extreme condition, right? They can tolerate the extreme condition. Now, what could be the extreme conditions? Again, in my earlier lectures, we discuss about the different, again, we talk physiological conditions because everything is based on the physiology of the cell. That was called as temperature, pH, right? Pressure, osmotic pressure. Then we have a certain nutrients. So we have certain chemical factors, some we have physical factors. So under these conditions, how the cells survive? If they go to the extreme conditions, for example, high temperature, low temperature, very low temperature. So if they deviate from the optimum condition or the median conditions on both the sides, both the extremes, we call it plus or minus, or for example, if we take about the pH, then it go to the total acidic or total alkaline, then such conditions are called the extreme conditions. So such bacteria which belong to them is called the extremophile, and this belongs to the group, if you remember, the domain Archaea or Archaebacteria. Mostly they belong to the Archaebacteria. Now, micro or macroorganisms which can survive in harsh conditions. So this is the definition, such as extreme high and low temperature, pressure, salinity, that is the presence of salt, etc., are known as the extremophile. Now, these are some pictures showing some, some hot springs, bacterial mat, golden brown in colors, right? Then we have, this is the geyser basin, and this is again we have extremophile on Mars, could survive on the hundreds of millions of years. Then this is the Pyrolobus fumeri, isolates from the deep sea vent fluid. So all these things, situations or these habitats are basically providing the bacteria to live in the very extreme condition. Now the question is how the bacteria survive in these habitats. The bacteria survive in these habitats after adaptation. And how do they adapt? They adapt based on the modification of certain component or certain parameters. Now look at this type of extremophile. This is the long list of extremophile. I think it's clear to you, visible on your screen. Thermophiles, high temperature. Hyperthermophile, very high temperature. See, up to 100, more than 100 degrees centigrade. Then we have a psychrophiles, low temperature up to -10, -20. Then acidophiles. We can grow these cells, acidophiles, even at less than 3 to 2 pH. Then we have a bacteria which can grow on the alkaline condition. We call alkaliphile. In alkaliphiles, the alkaline environment is important, right? High pH, it can grow on high pH. Then we have osmophiles, high sugar concentration. They can survive. They won't lyse, they won't shrink in the case of high concentration of a sugar. Osmophiles bacteria, they can tolerate. How they can tolerate? It is a modification of a certain structure, modification of a certain factors and parameters of the cell. Xerophiles, extremely dry conditions, they can desiccated environment, they can live. Then there's so many long list of these conditions, these extremophile conditions. You can go through it, like lithotroph, methanogens, and then endolith. All these conditions, they are posed by the environment and the habitat where the cells are living. But the cells can, from inside, they can produce the best possible protection against these things. These are some examples, examples of extremophile, thermophile especially. We have already discussed these in the list, and so again you can look into the methanopyruses, then you can have a Pyrolobus, and then you can have Escherichia coli in case of mesophiles. Then in case of psychrophile, you can have some bacteria which are called the different species and different genera which can survive at 0 degree centigrade to 15 degree centigrade. So basically all these examples of bacteria which can survive at a low temperature, at very high temperature, besides the mesophile which are on the moderate temperature of 37 degree centigrade, right, we can categorize them on the basis of certain physiology. Now again I'm using word physiology. Physiology means like a metabolic processes, biochemical reactions which are taking place inside the cell. Now if these physiological processes are blocked, are intervened with certain factors, are interrupted, then cell's survival frequency, cell survival status is being affected. Now let's talk about the modification in thermophile. Now as you look into the thermophile, we discuss in detail as a whole membrane, nucleic acid, proteins. We have seen in case of prokaryotes, we have seen how the structure of protein, what is the membrane compositions and what is the membrane structures, then how the nucleic acid looks like and where the nucleic acid, size of the nucleic acid, structure of nucleic acid, or the composition of the nucleic acid, which is more important. Structures is not important, but composition is very important. Means what are the content of this nucleic acid, right? In terms of like GC rich region or AC rich, AT rich region. So all these factors are basically responsible for the modification of these states. Now, if you look the thermophile, then long chain iso branch structure fatty acids are present. So it means there's a slight variation from the normal prokaryotes, mesophiles to the thermophiles or to the archaebacteria or thermophiles in the membrane modification. Now this membrane modification help to provide the more rigidity, right? High stability and rigidity, stabilize membrane, reduce fluidity, right? At high temperature. So it means when the bacteria are grown at a very high temperature, they can be protected through these modification. All right. Similarly, nucleic acid composition is changed, high GC content. So high GC content can tolerate the high temperature at the because of the GC bond. GC bond is a strong bond, it's a stable bond. They can have a high melting temperature compared to AT rich region. So GC content is more in nucleic acid. Then proteins are very important because genes are not, GC content is not much important, which is very small, one of the components, where the protein rich or protein conditions or protein types are very important. For example, high catalytic enzymes are present, right? More disulfide bond carrying proteins are present, right? Then smaller loops, compact N and C terminals are present. So it means these modified proteins basically are different types of protein. Unlike the mesophiles, they provide the temperature tolerance, and so the thermophiles are modified through these means and make the thermophiles more sturdy, more strong against the high temperature. Now, category metabolism, if you talk about the glycolysis is one, so enzyme upregulated, so respiration protein downregulated. So this is again the key behind the tolerance against the high temperature. So Krebs cycle downregulated, ATP, acetyl-CoA buildup, they're all downregulated. Then it affects like immediate energy of supply, the protection from the stress. So because high temperature means like putting a stress on a bacteria, and they want the bacteria not to grow, become bacteriostatic. But I think having these metabolic processes, having this change in physiology, right, the bacteria can survive under these conditions, and they can provide the better protection from these abnormalities in terms of like environment. They can cope up with the situation. So this is called a tolerance against the temperatures. So these metabolic processes or physiological processes help them. Now what are the other molecules like osmolytes, which stabilize the protein by balancing the charges between the in and outside the membrane to maintain the integrity of the membrane. Then polyamines, right, stabilize the DNA and RNA, just like a histone protein in case of eukaryotes. So because in prokaryotes, histone proteins are not present, And histone protein is nothing but they are maintaining the integrity of the whole chromatin and euchromatin state. So polyamines are present which bind to the DNA and RNA and protect the RNA not to RNA stem loop structures not to degrade or not to lost the structure. The small DNA binding proteins again SEC7 and SSO7 are very common, but they basically provide the compact DNA structures and then prevent the damage of the DNA. So at high temperature there is the chance of always to lose the DNA or to damage the DNA. So when the DNA is damaged, then the cell will not survive. No proper replication takes place, cell will be destroyed, genes will be lost, some essential gene must be lost. So in order to protect them, the cell adapt the conditions at high temperature by small DNA binding protein. Then we have a DNA repair proteins. Sometimes the mutations take place. So when the temperature is very high, temperature mediated mutations take place. Now these temperature mediated mutation or temperature induced mutations are basically maybe lethal, or bacteria may be lost. So in order to protect the bacteria, some repair mechanism get activated like SOS, right? Some other mechanism like nucleotide based excision mechanism, and then various endonucleases, part of the polymerases, they get activated. So all these changes takes place at the high temperature, which may not lead to the loss of the cells or affect the survival frequency of the cells in a particular environment or particular system. Now the small heat shock proteins, which are very, very important, these heat shock protein basically help to maintain the protein folding or protein structures and to maintain the integrity of the cell not to die. Now modification in thermophile, this is the pictorial presentation of some protein which are involved in like polyamines in the DNA, like DNA repair mechanism proteins, then DNA binding proteins, right? Then we have GROELs and to help in the misfolding of a protein, and then DNA K and GROES. So these proteins are basically help, and some of the proteins which are involved in the osmolytes, they can maintain the membrane and not to shrink or not to lyse the membrane under certain disbalance or imbalance osmotic pressure of the cell. Now the modification in the sacrophiles, how the sacrophiles are modified. If you remember in my earlier lecture, we talk about the different integrity, different structure integrity of these membrane, cell membrane, then we have cell wall, cytoplasm in the bacteria. Now here we talk about the modification in these factors, modification in these characters. Having the modification in these characters, it can be of harsh condition can be tolerated by the bacteria. For example, sacrophile. Sacrophile are those which can grow at the -10°C up to -20 to -10°C. So such bacteria which are required to grow a temperature at optimum temperature of the 0°C or the low temperature, they are modified their unsaturated fatty acid, methyl branch short chain fatty acid, high S2 or iso ratio. So all these modification compared to the mesophile or compared to the other bacteria, eubacteria, they adapt themselves as to grow in the different physiology or to grow them in the different conditions. Upregulation of a gene like desaturases, Cas2, Cas3, cis and trans isomers. So all these things can be changed, can be modified in order to maintain the survival of the bacteria. All right. Now when the bacteria survived by changing the physiology, it means the bacteria is adapting the new conditions, environmental conditions, in order to survive. For example, provide the rigidity and protect against the UV stress, membrane pigments. Upregulation of genes desaturases, promotes like synthesis, conversion of the adaptive lipids molecules, because they provide the strength for the cells to survive in the low temperatures and may not be lost the function. So whether it is a thermophile, sacrophile, or acidophile, or acid or salt tolerant bacteria, they all get modified. So if you compare the extremophile, including thermo, sacrophile, acidophile, compared to the mesophile or the eubacteria or the normal bacteria, you will find lot of variation, lot of changes and differences among the different factors and different entities, right from the cell membrane to inside the cell. Again, regulatory molecules like a compatible solutes, for example, mannitol, then betaines, then sorbitol, and then you have glycines, glycerol. All these things are involved in modification, which maintain the osmotic balance and lower freezing point. Because when the bacteria grow at lower temperature, -10, -20, even sometimes much less than that, then it might freeze. So these molecules or these regulatory molecules protect them from their freezing condition. So they can change the freezing point, right? They can further reduce the freezing point at high temperature. Ice nucleating proteins, we call I and P. Ice nucleating protein control the extracellular ice formation, right? So that they may not form the ice, direct crystallization outside the cell, right? So all these modifications help these bacteria to survive under the conditions which are different condition, which are the harsh conditions, are the different condition from the normal bacteria. Protein modification is very common. Higher Kcat Km value maintain the enzyme activity at low temperature. That is, so cyclophiles, they modify the Kcat Km by modification of a protein or enzymes and make it flexible but less stable. Then we have a loop content in proteins, right? Allow the micro unfolding flexibility, low energy catalysis. All right. Alanine, glycine, isoleucine, arginine, proline, leucine enhances the flexibility. So such kind of amino acids are present. So you can modify sometime. No, this is very important from the industrial application. And that's how the very extremophiles, sometimes they are very good for the industrial production, industrial product formation, or industrial product application, right? Certain enzymes can be identified. For example, Tech polymerase, we'll give the example, Tech polymerase, which is present in the very important reaction of PCR. Now, this PCR is basically nothing but it is amplification of the DNA, which required the high and low temperature fluctuation at different time interval. So when you go for the high temperature, DNA get damaged, but it is not like that. Replication continues at high temperature. So how? Because it comes from the enzyme which can tolerate the high temperature from the bacteria which are Thermophilus aquaticus, which lives in the hot springs, or which are thermophile group. So this is the advantage and application of these modified conditions. So you can artificially modified by side-directed mutagenesis, you can modify these proteins, you can modify these amino acids, or naturally they can modify it through adaptive mode of bacteria by changing the physiology. So this is the chart showing the bacteria and their survival pH. So at different pH, you can from 0 to 13, you can see that some of the bacteria they can survive at acidic conditions, some bacteria they can survive at the moderate conditions, some bacteria they can survive in the basic conditions. So these three category of bacteria, which we call the acidophiles, are extremophile, acid extremophile, or pH extremophile, they can have better opportunity, better physiology, modification, changes, adaptation under certain condition from low to high pH, and so we categorize them as the different acidophiles. What are the modification taking place in these acidophiles? Just like in thermophile and cyclophiles. So again, the membrane modification takes place by the tetra ether lipids are present with ether bonds instead of ester bonds. So this is the big modifications. So in this case, it is a ester bond, not ether bond, because ester bond will protect it from growing at the extreme condition with a high tolerance in the acidic condition, right? Then modification in the sugar moieties, for example, pore size, bioenergetics, then fluidity, fatty acid chain length, all these modification in these components taking place, which may maintain the membrane integrity at a low pH, right? Similarly, fatty acid isomerization takes place from cis to trans in case of acidophiles. This property adjusts the fluidity under the acid stress. So at the low pH condition, as acidic condition, these cells will survive because of these modifications, right? Then pH regulations and the membrane stability is also provided by the cyclopropane. Now cyclopropane provides the flexibility in the pH regulation. You can regulate the pH up and down by cyclopropane. So all these modification taking place as a as a type of mechanism or modification, a mode of action of these bacteria under low pH conditions, right, or high pH conditions, right, provide the bacteria to survive in the extreme condition. Protein modification, again, like altered surface charges so that they cannot affect by the low pH or high pH. Reduce salt concentration or salt bridges. It modified the active sites for the binding purpose of various substrate if it is enzyme or even a protein. So it contributes the acid tolerance. Protein pumps, it can kind of efflux pump, it can provide the best pumps, right? Then you have a transporters proteins. Now respiration linked protein transport coupled to ATP synthesis provide the energy and pH homeostasis. Now what is most important is pH homeostasis. From high to low or from low to high pH is balance in between called homeostasis. By modification of these intracellular or intercellular changes at the organelle, pseudo-organelle or the cytoplasmic level or pH membrane level changes in a pH extreme condition. So this change in this physiology adapt the bacteria, allow the bacteria to grow in the extreme conditions. So these modifications, so the question, if question comes like, what are the modifications and why the cells are being why the cells adapted in the extreme conditions? So the answer would be they adapt the conditions because of the modification in the membrane modification, protein pumps modifications and so on. Then chaperones and repair proteins are present. So there's many examples like HDEA, HDEB, HDEG, P, SIR-A, heat shock proteins and DNK proteins and several GROEL proteins. So a number of proteins, example of proteins are present which are modified, and these modified proteins are protecting the bacteria and protecting the folding and refolding and misfolding of a bacteria phenomenon to maintain the functional part of the protein under the extreme condition. This is most important. If this modification is not takes place, then bacteria will not. Suppose if you take the mesophile, in which there is no modification takes place. If you grow the mesophile at a high temperature, at high pH or low pH, then the mesophile will not survive. Why? Because these modifications are not present in the mesophiles. Yes. So this is the reason of physiological changes and maintaining the buffering conditions for that. Now then we have metabolic modifications, increase glycolytic rate, depends on again at low pH. It provides the ATP to sustain homeostasis in the high or low pH. Amino acid catabolism take place. Catabolism of endogenous RNA is taking place. Provide the ribosome base and energy. Polyamine accumulations, enhance acid resistance, breakdown of organic acid entering the cytoplasm, reduce proton load. So basically all these metabolic processes or modifications are basically taking place in order to adapt the conditions which are not a normal condition but away from the normal condition. What are the normal conditions? Normal condition, we call them conventionally as a condition which are required for the mesophiles to grow. That is the optimum conditions. So beyond the optimum condition, every condition is the harsh conditions. Biofilm formations. Basically biofilm formations are basically the best mechanism for the bacteria to protect them into colonize them to a particular colonies, are under the matrix of the lipopolysaccharides, are under the protective matrix, right, where the cell to cell communication take place and their cells guide to each other that not to be worried about it, we are safe. So such kind of signals are moving from one cell to another cell. Even the cells which are not belonging to the same species or even the same genera, they can form a biofilm formations we call mixed biofilm and provide the corrective resistance to acid condition or acid stress. The good example is the Tech polymerase discovery. Now the Tech polymerase discovery from the Thermus aquaticus in 1965, in 1969 Thermus aquaticus was discovered, and it was discovered by Carey Mullis later on as a PCR who got a Nobel Prize in later on. Now this is basically enzyme are bacteria present in the hot springs, right? Yellowstone National Parks, right? In 1971 and 74, identification of the thermostable enzyme from this Thermus aquaticus take place, right? And that was a breakthrough of the DNA polymerization, and that was the breakthrough of the DNA polymerase or the PCR reaction. Tech DNA polymerase described the Kane, Edgars, Trella, who purified the DNA polymerase 1 enzyme from Tech polymerases, and then later on it was used for the PCR reaction. They can tolerate at a high temperature up to 80 degrees centigrade. So the optimum temperature for this enzyme is about 70 to 75 degrees centigrade. What is the application of these extremophile? The application of these extremophile is like Thermus aquaticus, which is a Tech DNA polymerase. It's used in the PCR. High temperature stable enzymes for food, textile, detergent industries that are present. Thermostable enzymes for the biomass conversion, right? Biofuel production, catalyst stable at high temperature, pH, or solvent. So all these applications are possible only when these modification takes place in these at the gene or protein level. Then Psychrophiles, pseudo Alteromonas. Pseudo-Alteromonas is a species of the Psychrobacter, cold active enzyme. So because these Pseudo-Alteromonas is basically producing the cold active enzymes, lipases, proteases, right, amylases, and they are used in the detergent, food processing, and biotransformations, right, and at the low temperature. So when you required a low temperature where the normal bacterial enzymes may not work, but these enzymes work. For example, if you are working on some reaction at the cooling conditions, right, at the 0 degree or 15 degree, then you are not able to use the enzymes isolated from the mesophile, because mesophile enzymes can have optimum temperature of 37 degrees centigrade, and what you need is the cooling conditions at 20 degree, 10 degree or 4 degree. So for that the Psychrophiles enzymes are required, and they can be best used for the application or industrial application. Antarctic yeast and bacteria, anti-freezing proteins can be isolated. Since these Antarctic yeast and bacteria provide the anti-freezing protein, so these anti-freezing proteins can be used for the cryoprotection. So many times you need something to cryoprotect. For example, you are maintaining something like at below 0 degree, minus 80, minus 20, minus 40 degrees centigrade, then there is a chance of crystallization. So you can protect from the low temperature, damage from the low temperature. We call the cryoprotection, or such molecules or such agents are called cryoprotectant. Agriculture and molecular biology, it is well used in agriculture and microbiology. Cold adapted bacteria, Psychrophiles are bioremediations, they are used for bioremediations. Breakthrough of the breakdown of the pollutants in the cold environment, right? So all these applications from the extremophiles are good in industrial applications, and many applications for the purpose of the environment and industry. Similarly, acidophiles, you have various acidophiles like Helicobacter pylori. Again, it is acid tolerant bacteria. That's how it lives in the gut. It lives in our stomach where the acidic conditions are there. So they can tolerate there, and these bacteria can be exploited for like model for the medical and microbiological research, right? Then you have then you can have a dairy and fermentations, right? Various products which are fermented, you need the acidic conditions or low pH conditions. There you need to have this kind of acidophile. So basically all these extremophile and thermophile, psychrophile, acidophile are the bacteria belong to the archaebacteria or extremophile bacteria. They all have a very strong applications in the industry and environment, right? And they are good for the mankind's, right? Now when we use these bacteria, after you can also do some modification as per your requirement by site-directed mutagenesis, by protein modifications, by gene modifications, ultimately protein product development modification, and membrane proteins and membrane modification to provide the rigidity and reduce fluidity of the cells in order to maintain into the harsh conditions. Now, when you maintain these cells in harsh conditions, then you can isolate the metabolites, and these metabolites can be used for the same, at the same conditions, same temperature, pH conditions, for the better yield and better production. So I think this is one of the advantage of these extremophile which we have talked about. And so the take home message is that how physiology play a significant role in maintaining the microorganism in extreme climate, in extreme conditions, what are the factors responsible for these physiological changes, how they adapt in the different conditions, right? We learn all today. And the most important part, our take home message from this lecture is that how these extremophile or how these adaptive conditions can be make use of it, better use for the application in the for the mankind's in agriculture, in food industries, right, in medicine, in in plant of the kind of fermentations, right. So different industrial applications are very prominent. Applications are available for these use of extremophile. So we have to be very careful about the identifications, classifications, and their diversity, maintenance, and their classifying them into different groups for the recalling them with their characters which are used for the various application. Thank you very much.